Now let's talk about the RNA viruses, which are less straightforward than the DNA viruses. In contrast to the DNA viruses, RNA viruses all contain single-stranded RNA, except for the reovirus, which is double-stranded. Once again, here is another exception to the rule. And thinking back, do you remember what is the only single-stranded DNA virus? The only single-stranded DNA virus is parvovirus. Remember, it is part of a virus. And what is the only double-stranded RNA virus? It is reovirus. Very good. So as a learning strategy, it is important to understand the general concepts and then remember the exceptions to the rules. This approach helps us keep in mind the most important concepts while also appreciating that nothing is 100% one way or another. The RNA viruses can be either positive or negative sense, and they can take on one of three forms we mentioned earlier in the lectures. They could be naked or enveloped, icosahedral, or helical. Do you remember back to the DNA viruses, are they mostly icosahedral or helical? Remember, most DNA viruses are icosahedral. And what is the one exception to this? Remember, the pox virus has a complex protein structure that is neither icosahedral or helical. Very good. And another rule to remember is that all helical capsids are enveloped. Now we're going to discuss the difference between positive sense and negative sense RNA viruses. This is a very high-yield concept, and it can be a little confusing, so let's go through it slowly. Like we've said earlier, single-stranded RNA can either be positive sense or negative sense. In order for this RNA to be translated into protein, it needs to be in the proper form. So to make it simple, positive sense RNA molecules are mRNA molecules. Positive sense RNA molecules are mRNA molecules. This means that they are directly translated into protein by ribosomes, just like the messenger RNA is supposed to. So let me say that again. Positive sense RNA viruses are directly translated into protein, just like messenger RNA is supposed to. And where in the cell does this happen? Well, remember, translation is carried out by ribosomes that exist in the cytoplasm or on the rough endoplasmic reticulum. Very good. Here is a list of all the positive-stranded RNA viruses. We have the retrovirus, togavirus, coronavirus, hepavirus, calicivirus, and the picornavirus. A mnemonic in first aid to help us remember them is, I went to a retro toga party where I drank flavored Corona and ate hippie California pickles. Hopefully we can use this to remember that retrovirus, togavirus, flavivirus, coronavirus, hepavirus, calicivirus, as well as picornavirus are positive-stranded RNA viruses. In contrast to positive-sense RNA, negative-sense RNA viruses are complementary to messenger RNA. Therefore, before it can be translated, these RNA molecules need to be converted into positive-stranded RNA, and then they can be properly translated into protein. So let me say that again. Negative-sense RNA must be converted to positive-sense RNA and then translated into protein. Do you remember what enzyme would convert RNA to RNA? Is that something that eukaryotic cells or human cells can do? Convert RNA to RNA? Well, no, that's why it's so cool. Negative-stranded RNA viruses must carry with them an RNA-dependent RNA polymerase, a polymerase that uses RNA to make more RNA. Only these viruses do that. Now that is so awesome. So in review, negative-sense RNA is converted by RNA-dependent RNA polymerase into positive-sense RNA, which as we said is equivalent to messenger RNA, and then it is translated into its functional proteins in the ribosomes to form a complete virin. So in review, Negative-stranded RNA viruses must have or carry with them this RNA-dependent RNA polymerase to make the protein and to complete the virin.